Welcome to Genetics 311. We're going to talk about negative repressible gene regulation. So just to review, regulatory systems have multiple components. For example, they have the promoter region, which is a site for RNA polymerase to bind and start transcription. The operator region is where the regulatory protein binds to the operator to control the expression of the structural genes. We have the structural genes, which code for proteins. We also have the small molecule, which binds to the regulatory protein and determines the activity of the regulatory protein. In negative repressible regulation, negative means that the protein is a repressor protein. This means that transcription is repressed when bound to DNA. Repressible means that transcription is off when the small molecule is present. So to recap, the default state of gene transcription is on. In negative repressible, the regulatory protein alone cannot bind to the operator to turn it off without the small molecule. And so without a small molecule, the repressor can't bind to the operator, and so RNA polymerase synthesis continues or goes on. If the small molecule is present though, it binds to a regulatory protein and the resulting molecule complex binds to the operator and turns gene transcription off. So let's look at an example now. So we'll be looking at the TRP Operon. So just to give a little background information, bacteria like E. coli need amino acids to survive, and so they're able to synthesize many of them. Tryptophan is an example of one. It's an amino acid that E. coli can either ingest from the environment or synthesize using enzymes that are encoded by 5 genes. So right here we have the coding region, which includes the genes for the 5 tryptophan enzymes. We also have the promoter region, which I mentioned before is where RNA polymerase binds to initiate transcription. We also have the operator region, and along with that we also have the repressor protein, which binds to the operator region. We also have the small molecule, which is tryptophan. When there is a large amount of tryptophan, which represents a small molecule, the tryptophan binds to a TRP repressor protein at the operator sequence. This physically blocks the RNA polymerase from transcribing the tryptophan genes. When tryptophan is absent, the repressor protein does not bind to the operator, and RNA polymerase synthesis continues, and then the genes are transcribed. So once the genes are transcribed into a single mRNA, this is then translated to produce 5 enzymes to help E. coli survive. Thank you for listening to my presentation.